Welcome to Texel, a small island in Wadden Sea with a vast landscape of salt marshes and tidal flows. This unique environment is home to Salicornia europea, a plant that plays a crucial role in stabilizing coastal ecosystems. Join us as we explore its fascinating life and our journey to understand it.
Meet Salicornia europea, also known as pickleweed or zeekraal. It is a halophytic succulent that thrives in salty environments. But as economic activity drives up greenhouse gas emissions and sea level rise, prolonged flooding pushes Salicornia to its limits, with higher salinity, stronger currents, and shifting sediments. We're here in the Slifter in Texel to study how rising sea levels impact the growth and germination of Salicornia. From tides to salt levels, every factor counts in this delicate ecosystem.
Research on the effects of sea level rise on Salicornia europea is limited. This study will focus on how sea level rise influences inundation periods and salinity and how these changes affect the germination and early growth of Salicornia europea. Our hypothesis for the experimental design is increasing salinity and inundation times have a negative effect on the germination and growth of Salicornia europea. Our hypothesis for the observational design is with increasing distance from the gully, the Salicornia will have higher biomass and density and lower salinity. Our general research question is, what is the effect of increasing salinity and inundation times on germination and growth of Salicornia europea? To investigate this, we will have two designs, one observational in the field and one experimental design in the lab.
For our observational design, to observe Salicornia on a grady, we set up 3 transects with 3 quadrants, near a gully in the Slifter where Salicornia could be found. We collected clippings to dry back in the lab to determine the relative biomass of these patches through the dry way. We then collected soil samples to measure salinity and carefully logged our observations on the height and density of the plant. When we returned to the lab, we saved the clippings, cut them, and counted them. Once ready, we put them in the drying oven so that we could measure the dry biomass and compare the different variables within the quadrants.
In the lab, we germinated seeds under controlled conditions to test the effect of different salinity and inundation levels, replicating what's happening in these salt marshes due to sea level rise. To test the effect of salinity on germination, we used 6 salt plates and divided 100 seeds onto 5 cotton pads each. We wet the cotton pads using salinity solutions of 0, 15, 30, and 50 ppt. To test the effect of inundation times, we used a sieve tower, dividing 100 seeds onto 5 cotton pads on each sieve. Every sieve was given its own container, which we separated with each inundation time of 0, 2, 4, 6, 8, and 10 hours. The seeds are submerged into distilled water according to their specific inundation times. For both variables, we classified germination through a success or fail rate after 7 days.
Using the Kruskal-Wallis test, we found that our observation showed no statistical significance with distance from the gully and the other variables. We chose this test because of the small amount of samples that we had. No significance in weak relationship was most likely due to not enough samples and the other unexpected confounding variables, such as the puddle areas. We compared these areas with the other quadrants as they were inundated for longer, and we found that there was a lower biomass, density, maximum length, and higher salinity, which could indicate that longer inundation times negatively affect the growth of Salicornia.
Both experiments showed strong negative correlations with germination success. Each relationship was found to not be statistically significant, possibly due to small sample sizes, but we can still see a correlation between the variables, as seeds exposed to shorter water times and lower salinity had much higher germination rates, highlighting how both factors significantly affect Salicornia europea germination.
Of course, fieldwork comes with challenges. We struggled with our time management as the different inundation times would require us to be present when we couldn't be. Additionally, we unexpectedly came across confounding variables of different elevations and paddles into Slifter, and we didn't have enough samples to come to a significant conclusion. But every challenge was part of our adventure.
Our research into this topic has revealed that both salinity and inundation time have a significant relationship with the germination of Salicornia europea. While we can say that these variables have a negative effect on the germination of seeds, further research is needed on the effects on the growth of Salicornia. Though our investigation into the growth of adult Salicornia in the field did not give us as much information as we wanted, the knowledge that we have gained on the germinative behavior can help with the restoration of Salicornia in marshlands. Salicornia is more than just a plant, it's a vital piece of the coastal puzzle, and its survival depends on how we care for our changing coast. Let's protect these salt marshes and take steps towards a more sustainable future.